We're going to get started and talk about sort of cellular functions. Again, you hopefully had a chance to review the video on cellular basics that was posted in the content folder, and this is the lecture that goes along with our first day that you're going to watch over the weekend, or listen to over the weekend rather. Okay, so the cells in our body are basically called eukaryotes or eukaryotic cells. So all cells that have a nucleus are considered eukaryotic, so our human cells, eukaryotic meaning they have a nucleus and they have well-defined organelles. And you can see that there are organelles in this cell right in here, lots of them, so you can again review the basic parts both in your book and in the video that I posted. But again, for instance, you have ribosomes over here. Ribosomes are known for protein synthesis. You have lysosomes. Lysosomes in your cells are there so they can help digest dead cellular debris. You have mitochondria for making ATP, etc. So these organelles, all part of eukaryotic cells, all have very particular functions that they do for us. So all cells become specialized as they grow and mature. And this is something called differentiation. And I want you to remember that because you want your cells in your body to be well differentiated. This makes the cell more stable. So when you talk about cell differentiation, the more well differentiated the cell, the more stable the cell, and that's actually what you want. Once a cell is fully differentiated, it will always be that type of cell. If a cell grows up, for instance, to be a skin cell, it's always going to be a skin cell. It's not going to change its mind one day and decide to become a liver cell. So that skin cell will eventually replicate and make 2 new skin cells. Okay, so cells do a lot for us so that we can function. So there are a lot of cellular functions. What are some things that cells do for us so that we can actually function? Well, the first thing is movement. We have to be able to move around. So for instance, our muscle cells have to be able to generate enough force in order to help us move around. Okay, cells help us feel what's going on, right? That's called conductivity. So for instance, nerve cells have to conduct impulses in order for us to feel. So when that impulse is generated, the impulse has to be conducted somewhere, right? Meaning there has to be a path for that impulse to travel in order for us to feel the stimulus and then react to it. Okay, so our cells help us with that. Metabolic absorption, we need nutrients to live. I don't think anybody can really dispute that. All cells take in and use nutrients in order to function. They need those nutrients to make protein and to make energy, which is exactly what cells are made to do. That's what cells do in our body. They make protein for us and they make energy for us. Okay. They might need to secrete substances that can help us. So some cells make substances from other substances that they absorb. So they take in the nutrients, they break them down, and they kind of make substances, and then they secrete that new substance. A good example of this is mucus. So for example, when you swallow, you have goblet cells that are in your esophagus. Those goblet cells that took in nutrients make mucus for us. So as you're swallowing, those goblet cells secrete mucus in order to make the food slide down your throat a little bit easier, right? So some cells are very particular in what they do. All cells can rid themselves of their waste products. All cells have waste products because they take in nutrients, they break down, to use them. So they take in the nutrients, they break the nutrient down, and then what's left are waste products, right? So the lysosomes can help us in the cell, can help us with this, right? They either break down dead cellular debris and get rid of it, or they secrete or excrete waste products from the breakdown of some of the nutrients that they take in. Cells need oxygen in order to make energy. That is one of the principal things that cells do for us, is they make energy. All right, so cellular respiration is a term that you're going to hear again. So something you want to make a note of. Cellular respiration, basically cells take in oxygen and they use that oxygen to make ATP. And ATP, as you know, is energy, right? Adenosine triphosphate is energy. And they do this with the help of mitochondria. So the mitochondria help in cellular respiration. Cells have to grow and divide, right? So that is definitely something that they do. Cells divide and replicate in order for tissue to grow. Cells also divide in order to replace cells that have died. So almost all cells of the body go through a process called apoptosis. Apoptosis is just basically when a cell gets to the end of its life and it dies off. Well, something has to replace that cell, right? So the neighboring cell that's a little younger and maybe ready will go ahead and go through this cell cycle and replicate itself and reproduce itself in order to fill that gap where the old cell has died. And cells talk to each other. Cells love to maintain homeostasis for us. Homeostasis, again, another term you should be very familiar with. Homeostasis is that nice, steady state, nice even state, right? Called that our bodies really work to maintain. So cells will talk to each other in order to maintain that nice, even, steady state that helps us be healthy and helps us survive and function. Okay, so these are the cellular functions. They are all, again, listed in your book. You want to go ahead and review them there as well, all right? So, cells, some other things that cells that are sort of some things that you need to remember about cell basics, because remember, we're just talking about cellular function, sort of the foundation of cellular function. Cells need to stick together. Cells depend on an intact plasma membrane in order to stay together. This is vitally important, right? The plasma membrane actually has lots of different functions. You can see that I've put see table 1-2 on page 5 of your text for a list of the functions of the plasma membrane. Remember that when I put something in bold like that, I want you to go to that table and make some notes for yourself that this is an important table, and these are some things that you should remember. So, what does the plasma membrane do for ourselves? Well, if the cell didn't have a covering, what's going to happen to it? All the insides are going to fall out, right? So the plasma membrane holds all the contents of the cell inside the cell, right? So as it creates this barrier, then it also protects the cell from foreign invaders, such as bacteria and toxic substances, right? So in order to maintain the integrity of the interior of the cell, you can't let stuff in, right? So it acts as sort of a protection for the inside of the cell against all kinds of things that can get in. The plasma membrane allows for transport of electrolytes and proteins into and out of the cell. Remember, we just said that one of the things that the cell does is go through a metabolic absorption process, right? Well, how's it going to do that? If you have a plasma membrane that's keeping things out of the cell, how will electrolytes or nutrients get in? Well, they get in because this plasma membrane is very selective, and it knows exactly what to let in and what not to let in. Okay, so you can see that there are channels, membrane channels that allow certain electrolytes in. Sometimes the plasma membrane allows the cell to communicate with the cell right next to each other, right next to it, right? It has to recognize the cell that's next to it as a friendly cell so as not to destroy it. And sometimes it can help out if it needs nutrients. So if you have two cells side by side, they each have their own plasma membrane. In order to not destroy each other, they have to recognize that they are the same cell, right? And they have the same plasma membrane. So these plasma membranes will actually talk to each other a little bit. And if the cell on the right, let's say, is hurting a little bit, the cell on the left may be able to help it along because their plasma membranes talk to each other. They say, hey, I could use some sodium over here. So then the cell on the left will talk to the cell on the right, and the cell on the left will allow the diffusion of some electrolytes over to the cell on the right. So they do actually function together, okay? So you can see that the plasma membrane itself is made up of lipids, which are generally just fat. So they're made up of fats like cholesterol. They're made up of carbohydrates and proteins. That is what the plasma membrane actually looks like, and you can see that right here. So the lipids, the fats, are interesting because one side of the lipid layer likes water and one side of the lipid layer doesn't. So that this allows water to stay put and not diffuse out, right? There's water inside the cell, and that side that's inside the cell likes water, so it wants to keep water there. And the side on the outside doesn't really like a lot of water, so it doesn't want water to go out. Okay, so it'll also let other substances get in, like oxygen. So the plasma membrane, the lipid layer kind of functions like that. You also have proteins in this plasma membrane as well. Proteins are essentially just a chain of amino acids called polypeptides. These proteins are really important because they are essentially gatekeepers. They are the gatekeepers that recognize substances that are trying to get in and out, and they will bind to them and help them along. So for instance, they help electrolytes get in and out of the cell because they recognize that they're supposed to be there, and they have receptors for them, and they say, oh, yep, okay, you're good. Potassium, I like you, and I'm going to help you along, get in the cell. So they allow certain things to get in the cell and certain things to get out. They have cell surface markers as well that help them attach to neighboring cells in order to help maintain cell shape. So these proteins actually have some receptors on them, and they will bind to, let's say if they're on the right, they'll look at the cell on the left. Those plasma membrane receptors will attach to each other in order to help maintain that cellular shape. Okay. Now, some proteins, again, have these cell receptors that can recognize and bind to other molecules. Your book calls them ligands. So ligands are essentially a fancy term for a smaller, for a molecule that is smaller than the protein itself. So if you have a protein that's the size of your fist, the ligand is a little smaller than that. Okay, so that's really all a ligand actually is again a protein, or I'm sorry, a molecule that is a little bit smaller than the protein itself. Okay, so these proteins have these cell receptors, and it's like a lock and key. They recognize the substance and they connect with it. All right, so an example is a hormone. So I use this as an example. Hormones that usually float around your body only act on their target tissue, right? So thyroid hormone is always going to act on the thyroid, right? It's going to float around and it's going to look for the thyroid, essentially. When it meets the thyroid, like a lock and key, the hormone's going to fit with that plasma protein. It knows, oh, hey, I have reached the thyroid tissue. So the thyroid tissue with its receptor on it will take in that thyroid hormone, and like a lock and key, that will fit, right? The thyroid hormone's not going to go down to the liver and attempt to get in because there's no receptor for it there, okay? No cells have a receptor that's going to allow the thyroid hormone to get in, okay? When you get bacteria in your blood, your white blood cells, for examples, they like to greet it, and the antigens on the protein of the white blood cell with the same antigen on the bacteria, the white blood cell says, nope, I recognize you as foreign, right? So the bacteria comes along, and the white blood cell, which has, again, a plasma membrane that has these proteins with these receptors, looks at that bacteria and says, wait, you fit, I don't like you, I remember that you're not supposed to be here, and then the white blood cell will help destroy that bacteria, right? So it elicits the immune response, and that is how your body can get rid of bacteria. So again, these plasma proteins are really vitally important to the cell because they help the cell from being destroyed, keep the cell from being destroyed. Okay. Again, cells need a supportive extracellular matrix, right? So a cell cannot survive if it doesn't have some support to keep it together. Remember, a cell is just squishy, right? It needs that covering over it, and it needs something around it to either affix it to where it is or keep it, help it maintain its shape. All right, so cells are squishy. They need an intact membrane, and they need some support around it, right? So they need to be stuck to a cell on either side. So a cell by itself just isn't going to stay put. It's just going to flop around. So it needs a cell on either side. So cells that are grouped and stuck together actually form tissue. And tissues that are grouped together form organs. So you may remember this from anatomy. So, for example, your epithelial skin cells are nice and columnar, and you can see them right here, okay? So nice and columnar, these are your epithelial cells, okay? So they need these these epithelial skin cells are lined up. But if they weren't attached to something, they would just fall right off, right? So your skin would fall off if it wasn't actually attached to something. So it's stuck to this fatty layer right here called the basement membrane, okay? Now, this basement membrane helps keep the epithelial lining on top of, those skin cells on top. Okay, so the basement membrane is a sticky, intricate meshwork of proteins. It's embedded in gel. You can see it right here, okay, up in here in the corner. It's kind of, there's a lot of proteins embedded in like a gel substance. That's really, this gel is just made up of complex carbohydrates, okay? A lot of space in the matrix right up in here. You can see that it looks like there's a lot of space anyway. And that space is really important because substances can diffuse through it, like nutrients and waste products, right? So again, if you had a basement membrane that was sealed up tightly, nothing could get through it. The cells perhaps underneath, like the capillaries and all that, wouldn't get any nutrients. So it has to allow us to take in nutrients. All right, so it's really important. So this matrix is actually, this extracellular matrix is made up of something called fibroblasts. You can actually see the fibroblast right here. That's a fibroblast. Fibroblasts are these specialized cells that are only in the matrix, this extracellular matrix, and they'll make more if it's needed. All right. So this picture up in the corner is connective tissue, and again, it's called connective tissue because it essentially connects cells to make tissues and organs. So that's exactly what connective tissue does, right? All right. So that's the extracellular matrix. So cells require specialized cellular junctions in order to, again, stick together. Cells that are next to each other are often connected by these special junctions. These junctions not only connect the cell, but they also allow certain molecules to pass through from cell to cell. And that allows for very coordinated activities of these cells within a tissue type, right? So again, when the cell finally grows up, all cells of a particular, all cells in a grouping that are the same function in a tissue. So all cells want to do the same thing in that tissue type, right? So hepatic cells, cells of the liver, they're grouped together to make the liver. They all coordinate with each other and they all do the same thing. And a lot of times the cell, the molecules will pass from cell to cell, and these are very coordinated activities, okay? So there are some parts of the specialized junctions that you kind of have to remember. You have something called a desmosome, and desmosomes basically act to unite cells in a button-like fashion. So they're just little points that kind of work like a button. And so all they do is hold the cell together. They just unite the cell together. Like, I'm a skin cell, I'm a skin cell. And so they connect them together, and so they are attached. You have a tight junction. Tight junctions actually prevent the movement of substances and prevent leaking. So just like its name, it's a tight junction. If it's a tight junction, meaning nothing's going to get through it, it's going to prevent movement of substances and prevent leaking of things out of the cell, okay? Tight junction. You have a gap junction that are actually clusters of proteins. These clusters of proteins are actually called connexons. You'll see that word in your book, I believe. They're essentially just a tunnel that allows certain substances to pass directly from one cell to another. So again, if the cell on the left needs, a nutrient, then it'll maybe hopefully get it from the cell on the right. So there has to be a gap junction that's a direct cell contact so that the substance can go directly to the cell on the left when it needs it. So that's what a gap junction is for, right? Now cells actually talk to each other. This is called cell signaling. Cells need to communicate in order to maintain that homeostatic environment, right? That stable internal environment that they desperately want. They also need to talk to each other in order to grow and develop and organize themselves into tissue, right? Once cells start forming tissue, they go, hey, I'm a skin cell. Great, I'm a skin cell. I'm a skin cell. I'm a skin cell. They all want to talk to each other so that they come together, that they coordinate their function, and that they become the tissue type that they are destined to become. All right, so They do signal each other. They do talk to each other. There are lots of different ways that cells talk to each other. The first one is basically membrane to membrane contact. That's contact dependent. The membranes themselves, they're adhered, and they literally, the cells can communicate right through the membranes. Paracrine signaling is basically through the use of chemical mediators that act immediately. So the cell will release a chemical mediator, and it acts immediately on the body or on the cells. One of the examples that I can think of are histamine. You know, when you get a bug bite, right, or you get like a bee stings you, you get a bug bite or a scratch. Sometimes it becomes itchy. It becomes itchy pretty quickly. Well, histamine is what makes it itchy. Histamine, the cell secretes histamine. Histamine goes immediately to its target tissue, which is the surrounding tissue from the bite, and immediately produces an inflammatory response. Now we're going to talk about the inflammatory response in another couple of days, but that is an example of a paracrine cell signaling, right? And so it does that immediately. That histamine gets released, and you get an immediate response. You can have autocrine, which is secreting when the cell secretes mediators that only work on themselves. So that seems a little strange, I think, when you think about, well, why would I secrete something that only I can use, right? It's all for me. But cancer cells are the one cell that actually does this. Cancer cells are undifferentiated cells, right? A cancer cell is living among the differentiated cells, but it is not exactly like the cells next to it. It kind of does its own thing, right? So the cancer cell is a little undifferentiated, and it kind of takes over. Well, it signals, because it's undifferentiated, it sends out a substance, and that substance only targets itself, right? And cancer cell does this in order for itself to grow. Because the cell itself is undifferentiated, it doesn't really have a specific function except to make trouble. It kind of has to signal itself to be able to grow in a weird, irregular pattern, right? So it's why this is a function of autocrine. You have hormonal. Hormones can be released from cells. Hormones and neurohormones can be secreted to target certain cells of a particular tissue. I talked about the thyroid hormone again. The thyroid hormone is only going to work on thyroid tissue. That's where it's supposed to go. So hormones are a way that cells can signal each other. And then you have neurotransmitters. Some cells release neurotransmitters. If you remember from physiology, neurotransmitters are released at junctions called a synapse, right? So the neurotransmitter is released at the synapse. They diffuse, the neurotransmitter kind of diffuses through the synapse and jumps to the receptor on the other side to stimulate the cell. That's exactly how nerve cells work, right? So the stimulus has to travel along the neural pathway and then jumps to its receptor target cell, which causes either the movement or whatever the nerve cell was signaling to do. So again, cell signaling, cells have to talk to each other. That's what they do. They have to maintain that homeostatic environment, so they do that through signaling. Cells make energy for us. This is a huge piece of what cells do. Cellular metabolism, again, is a very important concept that you guys should really learn and think about. Cells need energy to function, and this is what cells do for us. This is called cellular metabolism. They absorb nutrients, they store them, they metabolize them, and later they use the stored metabolites to make energy. That's what they do for us. So cells make ATP. Again, adenosine triphosphate is needed. That is energy, and you can see it's really just adenosine with 3 phosphates attached to it, and that's what's considered to be sort of the energy that we need. ATP is needed for muscle contraction. It's needed for active transport of substances. It's needed for us to really survive, right? Cells that don't make energy anymore die because there's nothing to keep them alive. So how do we actually make energy? Well, This picture here, phase 1, 2, and 3 are actually in your textbook. So let's see if we can, this textbooks, pathophysiology in general especially, have textbooks with a lot of these crazy algorithms essentially, and some people love them and some people look at them and go, I don't even know what that's supposed to be. So I kind of like them because I kind of feel like it's a pathway that I can follow a little bit. So let's see if we can look at it together and figure it out. So in phase 1, essentially what this is telling you is that your body, we eat food, right? We all love food, love to eat, love to cook, whatever. So we digest food, right? That nice cheeseburger that you just ate, that Five Guys, big fan of Five Guys, go have a nice big bacon cheeseburger, and you have to digest that. So that nice cheeseburger gets broken down in your stomach into smaller pieces, right? So proteins get broken down into amino acids, and polysaccharides, which are complex sugars, get broken down into simple sugars, or what is considered to be monosaccharides, and fats get broken down into fatty acids. Right? So this is outside the cell. This is happening in our gut, essentially, and so the cell is then going to take in these smaller molecules, these amino acids, simple sugars, and fatty acids. That's what the cell is actually looking for. So it's going to take that in. So that's phase 1. Okay, so that we need to do that. We need to eat. We need, in order to make energy, we need to actually digest that stuff. So then you have the second phase. So the second phase actually occurs inside the cell. Okay, so phase 1 is extracellular, so that's outside the cell, and phase 2 is intracellular, all right, inside the cell. So the cell says, hey, we can use that stuff. So inside the cell begins to break down the items, okay? So the first thing that it tries to do is to break down the simple sugars, all right? So the cell says, we need this stuff. We need to be able to use it, and it begins a process called glycolysis. So phase 2 is essentially glycolysis. Glycolysis is the breaking down of glucose, right? Glyco, the Latin word, I believe, for glucose. So glucose is essentially a sugar, and the cell breaks down the glucose into pyruvate or pyruvic acid, so pyruvate, and then it breaks it down a little farther into something called acetyl-CoA. Now, acetyl-CoA, what you really need to know about that is that it's just an acid chain, okay? That's all it is. So you have pyruvic acid, it's just acids. Once you break apart the sugar, you're left with a string of acids, all right? So that is the second phase of the cell, cellular metabolism. The third phase begins something called the citric acid cycle. Now, if you remember from physiology, the citric acid cycle is actually called the Krebs cycle. I'm sure you all have heard that term, the Krebs cycle. So to make this very, very simple, the Krebs cycle is essentially just a series of chemical reactions, and you see that is what the citric acid cycle is telling you. It's just a series of chemical reactions that uses oxygen to break down carbs, fats, and proteins, okay? So remember, you had glycolysis to break down the sugar, but you have fatty acid that has to get broken down into acetyl-CoA. You have proteins that got broken down into amino acids. Those amino acids need to get broken down into acetyl-CoA. They kind of need to all get broken down even farther. So it does that through the Krebs cycle. All right. So again, the chemical reaction that uses oxygen to break down carbohydrates, fats, and proteins. The end result of that sort of Krebs cycle and sort of the whole process is the formation of ATP. So when you make the ATP, once you break down all those things, on the right-hand side, you build ATP. On the left-hand side, you have some leftover stuff, right? That leftover stuff is byproduct. That's cellular waste, right? Like, I have an N, I have a couple of H's, I have an O, so it forms the waste products that you see: sodium, ammonia, urea, water, and carbon dioxide, right? So the oxygen comes in, makes the ATP during the Krebs cycle, and then you're left with some byproducts. All right, so the mitochondria, actually, this is where this process occurs in the mitochondria, which again is the energy producing organelle in the cell. So the oxygen comes in. We know that cells need oxygen. That is called aerobic metabolism. So again, cellular metabolism, aerobic metabolism. Aerobic metabolism just means that it's using oxygen to do its job, okay? So if there is no oxygen coming to the cell still has to make energy. It's got to figure out how to make energy. If it doesn't have oxygen, it's going to revert to something called anaerobic metabolism. Anaerobic metabolism means that there's a lack of oxygen, and what happens is the cell starts to break down other substances to make the ATP. And what happens is that pyruvate, that glycolysis started, and you have that pyruvate that's way back at the top in phase 2. Well, in the absence of oxygen, it's going to break that pyruvate down directly, and that pyruvate is going to be converted to lactic acid instead of the acetyl-CoA and the proper pathway of the Krebs cycle. So the cell is still going to make energy. It's not as effective, and the byproduct is actually going to be lactic acid. Now, lactic acid is not a good thing. Cells cannot function on this for very long, all right? They will die eventually. The nice thing, though, is if you give the patient oxygen, right? If you feed oxygen to the cells, this process is reversible because the cell will immediately revert back to aerobic metabolism as long as oxygen's coming in, right? So one of the things that we will talk about, or you will talk about in med-surg, for instance, is putting oxygen on your patients. And it's not necessarily because they're short of breath. It's because their cells are starving for oxygen. And there are some symptoms that we will talk about later when we talk about shock and the inflammatory process, that you're going to see that your cells are starving for oxygen, right? And you can fix that process. It's reversible, all right? So that is really what you need to know about cells making energy, all right? They do it for us. They use aerobic metabolism. They need oxygen to do it. And it's basically just the breaking down of proteins, carbohydrates, which are polysaccharides, and fats, okay? So what else do our cells do for us? Well, cells, water and nutrients move in and out of the cell, right? We know this. This is, again, probably not a new concept to you. You probably studied this in physiology or in anatomy as well. Water and nutrients move in and out of the cell. So diffusion and osmosis, you remember those, fancy words there. That is considered passive transport. So you have passive transport, you have active transport, all right? So diffusion and osmosis are passive transport, all right? And if you remember, diffusion is very simply when solutes or particles go from an area of higher concentration to lower concentration, right? If I have more on the left than on the right, then I'm going to move from the left to the right. Particles are, okay? So the particles are going to diffuse from the area of higher concentration to an area of lower concentration. They're going to diffuse directly across that plasma cell membrane that we talked about. They don't need any help doing that. It's too crowded on one side. They got to move to the other side, okay? And so they will do that through diffusion. Osmosis, on the other hand, is the movement of water, right? So diffusion is the movement of particles, and osmosis is the movement of water. Osmosis is when water moves from higher concentration to lower concentration. Now, the plasma cell membrane has to be permeable to water in order for it to move, okay? So there are a couple things that you have to remember about osmosis. It's not just going to move on its own, all right? It requires the membrane to allow water to go in and out, and the concentration of solutes on one side has to be greater than the other in order for water to actually move. So if you have a lot of water on one side and a lot of solutes on the other, water is going to move towards that area of higher concentration of solutes, okay? So it depends on water pressure. It depends on water pressure, which is considered hydrostatic pressure, and it depends on solute pressure, which is oncotic pressure, okay? So solutes When I say solute, the word solute, I mean like electrolytes, plasma proteins, things within the water, right? Particles, they tend to pull water, all right? So you have pressures, and you have stuff, and you have water, okay? And water isn't just going to move by itself. It has to have a reason to move. So the water, again, is going to get pulled to the area of a higher concentration of solutes, right? So if you have a lot of stuff on one side and a lot of water on one side, it's going to go from the water side to the side with the stuff in order to even out the water content, okay? So I hope that makes sense. One of the terms that we use in nursing that you're going to hear a lot is osmolality. Now, you're going to hear that when you do lab tests on a patient, right? They might say, well, what's the urine osmolality, or what's the osmolality? You're going to look at that. So osmolality is what actually controls the movement of water between body compartments, all right? It's actually just simply a measurement of the concentration of molecules per kilogram of water. Every kilogram of water in your body, it's going to measure how much solutes is in there, how much stuff is in there. Plasma has less water, so the osmolality is higher than, say, the osmolarity. Now, osmolarity, I'm going to step back for a second. Osmolarity and osmolality are sometimes used interchangeably, but they are, in fact, something different. Osmolality is what controls the movement of water. So osmolality is the measurement of the concentration of molecules in water. Osmolarity is the concentration of the same molecules, but in a solution other than water, all right? So plasma is something other than water. Plasma has less water, so the osmolality is higher than the osmolarity since there is less water to measure. I hope that makes sense. So osmolality really should be the preferred measurement. We want to see what the osmolality is in water, okay? Not necessarily in other substances other than water. So although the words are used interchangeably, they are essentially different things. So always really look at the osmolality of stuff. You want to know what the concentration of particles is in water, okay? Normal osmolality of your body fluids is about 280 to 294 milliosmoles per kilo. So you should remember that, what the normal osmolality is. The other factor that goes into sort of water itself is tonicity. So if you look at this picture in the middle, you see that tonicity describes essentially how effective the osmolality of a solution is, okay? So how effective it is. So again, we're talking about the concentration of particles in water, and tonicity is important because you're looking for fluids that have the same or different osmolality than your body fluids, okay? So we talk about tonicity when we look at IV fluids. When you are the nurse and you go hang up IV fluids, you have to know what the tonicity is of the fluid in order to understand what's going to happen when you put that fluid into the patient's veins, okay? So when you look at hydrating a patient, you want normal tonicity. You want the same tonicity that's in your veins. So if your veins have an osmolality of 280, you want something that's around 280. So you look at an isotonic fluid, right? If you see on this picture, isotonic fluid is 285. Well, that's the same concentration as normal body fluid. So if I put isotonic fluid, which is essentially just sodium chloride, into my vein, what's going to happen to it? Well, it's going to stay there, right? If the vein is empty and I need to fill it up with something that is the same as what it's already should be there, then I put isotonic fluid in, and therefore it's the same tonicity as our body fluid, okay? We use isotonic fluids all the time in nursing to rehydrate patients, right? And to fill those veins back up to restore normal volume. Now, if I take a hypotonic fluid, you see it on the picture, it has 200 milliosmoles per kilo of water, then that's a hypotonic fluid, which means there's less particles than there are water. And you can see just in that cup that there's less particles than water, okay? So if I put a hypotonic solution, a solution that has less solutes than water, if I put that in the vein, what's going to happen? Well, water is going to go into the cell, right? Because now I have a certain tonicity in my blood. I have a certain osmolality in my blood. I'm going to put something that has more water in it than particles. The water isn't going to stay there. It doesn't have a reason to stay there. It's going to fill the cell so that the cell, which is a little drier, so it's going to diffuse out of the vein and it's going to go into the cell because the cell's a little drier, has more particles now than the vein I just put this hypotonic fluid in, right? So I might choose that fluid, like as a physician, to say, well, the patient's cells need water, and I need to put water back in them, so therefore I might choose a hypotonic solution. A hypertonic solution, all the way on the right-hand side, is 400 milliosmoles in water. So therefore, there are more particles than there are water in a hypertonic solution. So again, more solutes than water. If I put in my vein, which is normal, supposed to be like normal, 285 milliosmoles, if I put something that has a lot of stuff in it, a lot of solutes, what's going to happen? Well, it's going to take water that's inside the cell and it's going to draw water in, because it's going to create that oncotic or osmotic pressure that needs to, and it's going to pull water in, because now when I put the hypertonic solution into my vein, I am going to have more solutes than water, and I'm going to need more water, so the water that's in the cell, then I'm going to pull it out. Now, this is a great fluid to use when I need to pull water out of the cell. Let's say your patient has a ton of edema, and they are having trouble with that edema. We might use a hypertonic solution to achieve pulling water out of the cell, because we actually want to pull it out of the cell, and we just use the fluid to do that, okay? So tonicity of your fluid is very, very important, and the reasons why you need isotonic, hypotonic, and hypertonic solutions in nursing is going to be very, very important for you guys to remember, okay? So that's passive transport, just water and particles moving in and out of the cell without any real help, just changes in pressure and changes in concentration. On the other side, you have active transport. Sometimes you have an inactive transport. So mediated transport or active transport means that the substance trying to move across the membrane needs a little help, all right? And you see this when either the particle's too big and it needs a carrier, like a transport protein, to help it get through the through that plasma membrane, right? Because it just can't get through on its own. It needs something to help it through. That's active transport. You also see this when you're trying to get across a cell membrane, but there's a force that the particle has to overcome, right? So it needs energy to do that, right? So a lot of times ATP is needed to transport particles across the cell membrane. Not necessarily a protein for transport, but this is actually energy. You see this with electrolytes a lot. This happens when you are working with the sodium-potassium pump, where you see sodium move out of the cell and potassium move in. They are both positively charged ions, so they don't just move on their own, right? Think of two magnets together. When you have two magnets together, I love when my kids play with magnets. You have those two magnets together and the magnets won't stick, right? Because they're the same polarity, so they go left or right or up or down, and they try really hard. You try really hard to get them to stick, but they just don't quite. Sometimes you have to flip a magnet over to the negative end, right? This is the same thing. When you have two positively charged ions, they have to get across that cell membrane, but they're opposing each other. So it needs energy to basically overcome that and just pull it across, okay, and allow it go across. And that's really what active transport is all about, okay? All right, so water and nutrients moving in and out of the cell, yet another very important function of the cell. Now, cells create something called membrane potentials. again, we are just talking about all the cellular basics. We're kind of getting this sort of foundational stuff out of the way. These are things that the cells do for us in order for us to function as humans. All right, these are again our eukaryotic cells doing these things for us. So membrane potentials are something that a cell will generate. Again, with sodium and potassium, I use that as an example because this is the most frequently seen example of when you talk about membrane potentials. Now, this may be something you remember from physiology, that resting membrane potential. So with sodium and potassium, there always needs to be a balance because they are positively charged ions. There has to be a balance. And it's usually 3 sodium out and 2 potassium in, all right? And so when you have 3 sodium go out of the cell and 2 potassium go in, this exchange creates a membrane potential. You see this in your nerve cells. You see this in your muscle cells. Muscle cells and nerve cells function on membrane potentials, okay? So that's what they do. That's what generates the ability for us to get up and move. All right. So cells, so when the ions are there, they're supposed to be, when the ions are where they're supposed to be, and you see that there is supposed to be sodium outside the cell in the extracellular fluid, and the potassium is supposed to be inside the cell. When they're in the right spot, when you have more potassium inside and more sodium on the outside, that creates a resting membrane potential. So the cell is at rest when both the ions are in the right spot. Now, when the cell receives its signal to move, right, there's that neurotransmitter, there's that stimulus that says, I gotta move, I gotta make all these muscle cells move, right? Sodium will move into the cell quickly. When sodium rushes into the cell quickly, an action potential is generated, all right? And you see right here, this action potential is generated, and so that causes something called depolarization, all right? So this movement of sodium into the cell causes the cell to depolarize. The cell itself then is more positive until it reaches a threshold where it can no longer be positive anymore. It's like, okay, I'm done. And then once that threshold is reached, the sodium channel gate actually opens, sodium floods into the cell, and it pushes potassium out, and then it does that until the stimulus stops. And then what happens is the sodium gates snap shut and the potassium gates open, and potassium moves back in the cell and sodium moves back out, and the membrane potential actually begins to return to its resting state, and that's called repolarization. All right. So again, here at the resting phase, you have the cell, you have the sodium and more sodium outside, more potassium inside, and that just causes this sort of steady state. What begins to depolarize the cell is sodium rushing into the cell, or sodium entering the cell. Once it begins to be a little bit more positive, the sodium gates will open right here, and during this phase, sodium gates open, sodium rushes into the cell, potassium jumps out. It's like an immediate reaction, and then the sodium gates close, the potassium gates open, and during that time frame, sodium goes back to where it's supposed to be, potassium goes back to where it's supposed to be, and you kind of have that reverse process, and the cell begins to repolarize and then go back to its resting state, okay? That generates an action potential. You see this in cardiac muscle cells. You see this in skeletal muscle cells. You see this in nerve cells. This is how cells function in order to make your heart contract, in order to make your body move, in order for nerves to transmit impulses. So this is a membrane potential, and some cells do this, okay? Lastly, cells divide and grow. Cells do not last forever. They undergo what is called natural apoptosis. They die off, and then other cells are left to divide and replace those that are lost. They do this through mitosis. So mitosis is essentially the division of the nucleus, and they do this through cytokinesis, which is division of the cytoplasm. So again, inside the cell, 2 things have to double and divide. One is the nucleus, and the other is the cytoplasm, okay? So before the cell can actually divide, it has to double in size and duplicate everything on the inside, all right? And that's in essentially prophase, or in the early phase, okay? So this is essentially the cell cycle that cells will use. So first it has to grow and double in size and duplicate everything inside, and then when it splits, it should split into two complete cells that are identical. All right, there's 4 phases to the cell cycle that I really want you to remember. The first one is the S phase. The S phase is when DNA is synthesized. So again, you have to, in order for your cell to double and grow, it has to recreate its own DNA, right? So in the S phase is the synthesis of DNA. The next phase is the G2 phase, or what is called the gap phase. Okay, so the gap phase is important because that's when RNA and protein are synthesized. All right, it is called the gap phase because it fills the gap between the S phase, which is the first phase, and the M phase, which is the third phase. So that's the only reason why it's called the gap phase. It's like, well, your body has to have something to do in between these two important phases, so let's call it that. The M phase is the third phase. That's mitosis. All right, that's when the nucleus and the cytoplasm actually divide. All right, so that's the M phase. So the S phase is the synthesis of DNA. The M phase is the mitosis phase, and the gap phase is what occurs in between. The last phase is the G1 phase. That is the time between the M phase and the start of this DNA cycle all over again. Okay, so that is the essentially all the phases. Now, this process takes about an hour. These 4 phases, it takes about an hour to do that. So your cells can replicate that quickly, all right? So as far as cellular growth and division, that is really what I want you guys to remember, are just what those 4 phases actually represent. So the last thing I want to talk about is tissue. Now, we said earlier in the beginning that that cells actually differentiate into alike cells. So well-differentiated cells that are alike form tissue, right? So all skin cells are alike, all liver cells are alike, all nerve cells are alike, all astrocytes are the same, all skeletal muscle cells. So because they are well differentiated, they look like their neighbor cells, they form a tissue, okay? And groups of tissues then form an organ. All right, so there's 4 basic types of tissue that I want you guys to really become familiar with. And box 1-3 is 5 pages long in your textbook. And this box, which is page 25 to 30 of your text, has a list of all the different types of tissue and what goes into those tissues. So I do want you to become familiar as to what tissue actually fits in what category. So I'm going to go through a couple of them. There's 4 basic types. You have nerve tissue, you have epithelial tissue, you have connective tissue, and you have muscle tissue. Okay, these probably aren't new to you. You probably remember this from anatomy. So epithelial cells become epithelial tissue. That is groups of similar cells that are, again, grouped tightly together. Now, epithelium has a protective function, all right? So epithelium, you can see there's lots of different kinds of cells that are types of epithelium. You have simple squamous, cuboidal, so they can be square, they can be tall and columnar, they can be flat, but essentially epithelial tissue is protective, okay? It's what you find in the lining of on the outside of your body. So your skin cells are epithelial cells. You find it on the interior body surfaces of organs. So anytime an organ's surface communicates with the outside, then you will see epithelial cells. So your GI tract, right, your esophagus, your urinary tract, those all communicate with the exterior, right? Your esophagus communicates with your mouth, your oropharynx, your urinary tract also has direct access to the outside, right? So those are lined with epithelial cells. Epithelial cells also form glands like your thyroid and your pancreas. They make up functional cells of organs that have any kind of a secretory or excretory function, like liver and kidney cells are called epithelial or part of epithelial tissue. And if they have a secretory function, they're actually called parenchymal cells, but they are epithelial cells. Okay. Now, they can be, again, flat, or square. They can be cuboidal, like cubes. They can be columnar, tall and narrow. They can be single layers, double layers. They can be stratified. There's lots of different ways that they can be laid down in order to create the tissue. So all epithelial cells are supported by a basement membrane. You'll see this thin basement membrane right here. This is actually epithelial tissue. And so you see that they are always supported by a thin layer called the basement membrane. Epithelial cells are always firmly anchored in place by a basement membrane. The interesting fact about epithelial cells is they have no blood vessels, okay? They are nourished purely by diffusion of nutrients from the capillaries located underneath the basement membrane, right? So they are supported by that. They don't have their own blood vessels. Now, I have a picture here of what is called endothelium. Now, this is the inside of a vein. Endothelium is found in the lining of your heart, blood vessels, and lymph vessels. Heart, blood vessels, lymph vessels. It's called endothelium. They are they're classified as epithelial, but they actually function a little bit more like connective tissue. I'm going to talk about connective tissue in just a minute, but you do have to know that they are considered to be epithelial cells. Mesothelium is found in the linings of your pleural space, your pericardial space, and your peritoneal cavity. And it basically just because of where they function like connective tissue, but they look like epithelial cells. So that is why they are classified as epithelial tissue. Connective tissue have a huge role in our body, and so box 1 through 4 on page 27 through 29 has all the different types of connective tissue and what they do. Connective tissue essentially support organs. They provide structural framework for the body. They connect body tissues together and store energy. So again, they get their name connective tissue because they connect tissues to organs. All right. So connective tissue is is made up of collagen fibers. It's very strong, very flexible. It's also made of elastic fibers, which help it stretch, and it's made up of something called reticulin fibers, which also are thin and delicate. So it can be made up of collagen fibers, elastic fibers, or reticulin fibers. So again, it can be connective tissue that's very strong and flexible. It can be elastic in nature and stretch. It can be very thin and delicate. It just depends on where it's found. But it's called connective tissue because it connects tissues to organs. It can be loose or dense. Loose connective tissue you'll find under your skin. That is your subcutaneous tissue that you're used to feeling. When you pull up your skin, you feel that connective tissue underneath. It can be dense in as well. You find dense fibrous connective tissue when you're talking about ligaments and cartilage and tendons. And let's see, it can be elastic. Elastic, like the lining of blood vessels, and like your trachea and bronchial. So again, those are called endothelial cells, but they function like connective tissue. So that's you'll see that in blood vessels. Reticular connective tissue is what you see in the liver and spleen, and you have adipose tissue, which is loose and fibrous. That's your fat cells right here, and fat cells store energy, so you'll see your adipose tissue right here. Okay. Muscle tissue can be one of 3 kinds. You have skeletal, cardiac, and smooth muscle. Muscle tissue basically contains actin and myosin. They are arranged in these nice parallel bundles, and they aid in muscle contraction. So you can see how different muscle tissue looks from, let's say, connective tissue. So here's a nice bunch of fat cells. They're just squishy, loose, connective, fat right there. Whereas muscle cells, as you can see, are these nice, very compact, columnar in nature, just a really nice, completely different looking kind of tissue. Skeletal muscle is voluntary muscle. So voluntary muscle means that it's under conscious control, and you see it's striated in nature. So this actually is skeletal muscle you're looking at right here, right? These are sarcomeres right here. They're bundled together in this very, very perpendicular pattern. I'm sorry, parallel pattern right here, not perpendicular, parallel pattern right here. Skeletal muscle is anchored by tendons to bones, and this is what moves our skeleton and helps us maintain our posture. So this is what skeletal muscle is what helps us move. Smooth muscle is considered muscle tissue as well. Smooth muscle is involuntary, right? This is not under our conscious control. You will find smooth muscle in the esophagus and the stomach and intestines, right? Bronchi, the uterus, the bladder. So those organs contract and constrict and contract and constrict and get bigger based on what the needs of the body are. We don't make them do that. They're under our unconscious control. There are stimuluses that make them do that, right? They dilate. So again, it's involuntary. Cardiac muscle looks like striated muscle, but actually acts like smooth muscle, and it's only found in our heart. That is the only place that the cardiac muscle cells will be. Again, not under conscious control, so it acts like smooth muscle, but it looks like skeletal muscle, right? We don't have any control over our heart rate. It just does what it needs to do for us, and that's only found in the heart. And lastly, you have nerve tissue. So nerve tissue is found in the nervous system, the brain, the spinal cord, and your nerves. Very specialized cell tissue to react to stimuli and then conduct those impulses to various organs in the body. And they do that in order to bring about a response to a particular stimulus. Okay, so nerve tissue looks very different than all the other kinds of tissue. Neurons essentially will transmit that impulse, so you have neurons in the nerve cells that transmit the impulse, and you have neuroglia, which protect and provide nutrients to the neurons themselves, right? So this that you're looking at is a picture of an astrocyte, okay? Astrocytes are the most abundant glial cells, right? They anchor neurons to blood cells. So these glial cells actually will provide nutrients to the neurons or the nerve cells themselves. They help recycle neurotransmitters that get used during different synapses. So they're very, very important astrocytes, right? You also can see oligodendrocytes. They surround individual nerve cells in the central nervous system, and they form the myelin in cell membranes that basically give rise to the sheath that surrounds and insulates the axons of the neuron, okay? If you remember from anatomy and physiology, those are called Schwann cells in the peripheral nervous system. So again, in order for nerve cells to be able to transmit the neurotransmitter that makes them work, they have to have not only a receptor cell, right, or the neuron itself, but they have to have a protective cell that helps get that neurotransmitter, helps that synapse along the nerve junction, right? So that's how nerve cells essentially work. We are going to talk a lot about these different cells in the body when we talk about specific organs, like when we do neurovascular alterations. We're going to revisit what this nerve tissue is and what these nerve cells do. When we talk about cardiac, we're going to revisit what cardiac muscle cells do. And when we talk about blood vessels, we're going to talk about what those endothelial cells actually do. So it's going to be really important for you guys to remember what cell type goes in what tissue. What kind of cell are we talking about? Are we talking about connective tissue? Are we talking about nerve tissue? Because they all have the same function, but they all work a little bit different to make that function happen. Okay, so I hope that this makes sense. And again, this would have been something we would have covered in class, but I'm glad that you have the opportunity to at least hear the material. And if you have any questions, you know how to get in touch with me, and I will see you guys on Tuesday.